Well, our research is really going down to the basics. So it's a long title, but to make it short, we are really looking for new drugs how to treat cancer. And also this is kind of a long shot. There are several reasons why we do it and why we are doing it on Guam. So what I'm doing with my students in the lab is we're actually looking at the ocean. And why do you think we're going to look at the ocean? Well, the coral reefs have been compared to tropical rainforests in a lot of ways. They are very diverse habitats where a lot of organisms over here compete for a very limited space. And one way they do that is they produce chemicals to either overgrow other organisms, to be defended against fish feeding on them. And one way they do it is by producing these chemicals which are noxious or cytotoxic or have other effects. And so the coral reefs, rightfully, have been compared like similar to the rainforest as a pharmacy of the sea. The drugs from the sea is kind of the concept we are following. And one organism we have focused in especially are sponges. Sponges are one of the simplest multicellular organisms. They've been around from before the dinosaurs even. And these sponges are very interesting because they also harbor a lot of associated microorganisms, bacteria. Up to 40% of a sponge can be bacteria. And these bacteria are often symbiotic, living inside the sponge, and they're also contributing to the sponge chemistry. And there have been cases now where we can actually say that the bacteria living inside the sponge are actually producing these chemicals which have potential to be used as new anti-cancer treatments. So what we are doing now is we focused on Pacific deep water sponges which have been largely unexplored, and that means we are looking at sponges from a depth of around 100 feet to like 300, 400 feet down. And using a commercial diver here from Guam, Jim Pinson, and mainly around the Rota Peninsula, some of the dive sites where he goes down and brings us back the sponges. And then with these sponges, we go back into the lab, and what we are doing is, here are some of the sponges, we then grind up the sponge, and then we have a mixture of sponge cells and bacteria, and we plate it out on these agar plates, and the bacteria start growing. Then we pick these bacteria and try to isolate pure cultures of these bacteria, as you can see over here. And then we put some of these in the deep freezer for later use. And then we also use these bacteria and try to identify using molecular method, not just PCR and sequencing, to identify what bacteria we are dealing with, what are the species of bacteria we are working with. And then later on we grow up the bacteria and we look at the chemistry. What kind of chemicals are these bacteria producing? So just some numbers. So far we have found about 70 sponges, and then it shows the enormous biodiversity we have in Guam. 11 of them are considered new species of sponges. We have isolated about 650 colonies of bacterial strains, and again about 43 isolates are potential new bacterial species. It just shows the enormous potential of biodiversity we have around Guam, and which hasn't been really explored, neither taxonomically or from the chemistry standpoint, to produce new interesting chemicals. These are just to give you an overview. We have really colorful bacteria. That's kind of the scientific names you get after we sequenced them and compared it with databases available on the internet. And these bacteria, we grow up and then we test them to see if they produce interesting chemistry. So far, we cultivated about 27 strains, and 6 of the extract from these 27 strains showed anti-cancer activity. And we are following now up what the active principles in this mixture of chemicals is. So these crude extracts can be anywhere from like maybe 10 to 50 different compounds. And then we have to go to certain isolation steps to find the pure compounds and then test if they are responsible for the activity we observed early on. We do the same thing with our sponges. Here just some of them, as you can see. Then we extract them just to show you how that works with solvents. You get sometimes really colorful extracts. Then we purify these extracts with column chromatography. We separate them out between a liquid and a stationary phase. until we end up with the pure compounds over here. It's the chemical structure of pseudoceracine, which we isolated from a marine flatworm. And once we get the pure compounds, we go back to our initial screening results and test if these compounds are responsible for the activity we observed in the food extract. So out of the U56, we kind of just last fall published a paper that we could actually prove the concept that these deep water organisms really have great potential as producing interesting chemicals. So over here, that's from one sponge. We fractionated it out in 16 different fractions, and then all of these were tested with all these different cellular assays. So we have these, we're looking at processes inside the human or cancer cell, and then these processes, what we are looking for is pretty much in the cancer cell, they are overexpressed, so we are looking at chemicals which inhibit these processes and suppress the cancer to grow or to proliferate further. And with these results, we could actually show that our idea of following up this enormous biodiversity is right. We got a hit rate of 47% of our sponges show activity in these assays, whereas other people looking at sponges from everywhere, they only had a 9% hit rate. So it just shows that the Guam and the deep water Guam environment is really special, and we have a unique biodiversity which we can explore over here. That's what we like to continue with the next grand application. These are some of the compounds which are recently isolated from, that was a sponge Suberia species. We don't even know what Suberia sponge it is, so it's a new species as well, this sponge. And we isolated this area of compounds, and they all look very similar, and they just differentiate, like over here you have a hydroxide group, or here another bromine. There are fairly small differences in the chemical structures of these compounds, but these small differences kind of pronounce differences in the activity against cancer cell lines. So that's what is some of the research that's still ongoing at the moment. Outcomes, we published 4 publications already on our work, and they are available online if people want to read up on it. We gave presentation at a total of 8 national and international conferences about this work, and we still have several publications in the pipeline where we're going to report progress on our ongoing work. Another important outcome of this, all this work is only possible with students which come to our labs and then which we train in these methods. So we had 3 graduate students over here who were working on these projects, and also quite a few undergraduate students, which are still, some of them are still at UOG, some moved on to Loma Linda University. Ron Paulina did a master's with Dr. Leon Guerrero, and then continued now with a PhD program. And I also had several interns from Guam. They came back for the summer holidays to Guam. They are from Guam, but they were working at different universities in the States, and they contacted me via email, said, "You're doing interesting stuff, could we work in your lab?" I said yes, and I trained them. And some of them are now going on with a PhD in chemistry at the University of Mississippi, and Syra actually got accepted at the University of Michigan Medical School, and she wants to come back to Guam once she completes it. So I think this is one of the major outcomes for this research as well. It's not just that we're discovering potentially new anti-cancer drugs, but we're also training our local students over here in biomedical research and how they can develop this into a career that can provide them with a future career, and also come back to Guam and work on the island to improve the health environment on Guam. So finally, I would like to thank all the granting agencies which have enabled this research to go forward, and I would like to thank you for your attention.